Hello. Okay, so this is the first of two sessions on AI in structural biology. This first one is going to be focusing on predicted models and how to use them. For our first talk, we have Eva Smorodina from the University of Oslo, who's going to talk to us today about AI for molecular modeling and protein design. So take it away.
Thank you for the introduction, and hi everyone. Since I'm starting this session, I'd like to provide an overview of available AI models for molecular modeling and protein design, and I will try to make it more practical and general so you can understand what types of methods exist and what types of methods you can actually use in your work. Here you can see a general overview of the talk for today. We will cover a bit of a theory about structural bioinformatics and molecular modeling, some old school methods or physics-based methods, then we transition to AI-based methods. I'll talk a bit more about the difference between structure prediction, inverse folding, and protein design, and then we will look into specific cases and specific models that you might use in your work. So let's start. What is structural bioinformatics? Probably you might know it already, but it's an interdisciplinary field. So it combines biology, chemistry, math, computer science, and sometimes physics. It helps you to analyze models and predict the models of biomolecules. Overall, the main idea behind molecular modeling is to help to understand the connection between sequence, structure, and function. Sequence, structure, and function is a part of the central dogma of molecular biology. We know that sequence defines structure and structure defines function. It means that to fully understand how molecules work, their function, we need their structures. So currently existing experimental methods like X-ray, cryo-EM, or NMR are time and resource consuming. That's why they're not always available for all the people. And we need molecular modeling to overcome the challenge of experimental methods. For example, molecular modeling can address the issue with much more sequences and much less structures. With molecular modeling, we can model the absent structures of those sequences that are currently existing. On the slide, you can see that there are many different types of molecular modeling tasks that address very different types of systems. You can address drug design problems, protein structure prediction, personalized medicine, and work with proteins, RNA, DNA, and many other systems. But today we will focus only on two of the tasks for molecular modeling. It's protein structure prediction and protein design. And by the way, the Nobel Prize in Chemistry in 2024 was awarded for protein structure prediction and computational protein design. So both of these topics are very important and trendy right now. So structure prediction is a computational method that allows you to get a 3D model of a biomolecule using only their sequences. For example, if you want to get a structure of industrially important protein that can be potential therapeutics, but you cannot get their structure in other ways. The second task is protein design. Protein design is a bit more general term that involves various modifications or engineering of existing proteins or creating new proteins that address specific tasks. For example, one of the tasks can be creating a binder that will target a bad molecule, so creation of potential drug. Prior to AI, also known as data-driven methods, there are multiple physics-based methods that address protein structure prediction problem. Three most common of them are homology modeling, threading, and ab initio folding. I will briefly go over them. So homology modeling relies on the principle that similar sequences should have similar 3D structures. It derives from the evolutionary information about proteins. Threading is quite similar term, but is different in terms of sequence similarity. So in trading, we don't have to have homological sequences or evolutionary close sequences, but we should have similar folds. And then we are able to try to insert or fit the sequence of interest in the available fold as a template. And ab initio folding overall is a holy grail of all types of protein structure prediction because it assumes that using only general biochemical and physical laws, you can predict the structure of biomolecule just from a sequence. Complexity of the task grows from homology modeling to ab initio folding, and now scientists shift toward AI models, though the ideas and general principles behind AI models to addressing structure prediction are similar to the principles in homology modeling, trading, and ab initio folding. For example, famous AlphaFold3, the model that is able to predict protein, and actually not only protein structures, relies a lot on evolutionary conservation, which is also used in homology modeling. In terms of trading, there is a model called protein MPNN that generates and fits a new sequence into existing fold, which is actually quite similar to the trading approach of fitting your sequence into existing template folds. And ab initio folding is very similar to generative structure prediction when you try to generate the complex or a molecule at all alone. This way, we get three concepts: protein structure prediction, inverse folding, and computational protein design. Let's talk a bit more about the differences between these three approaches. So structure prediction is when we move from sequence to structure. We want to predict the structure of a given sequence. Inverse folding moves in other way. We have a structure and we want to find a sequence that will fold into this structure. Protein design can go in more ways. For example, you can have a sequence and you want to redesign it to make another protein. But you can also redesign the initial sequence to get a structure at the output. Also, you can start from a structural template of the existing protein and redesign it to get a novel sequence. Or you can redesign the structure and get a structure as output. I hope it's clear because it will be useful for the next steps. For protein design, you can generally divide into the three different categories: rational design, fragment-based design, and de novo design. Rational design involves small changes into existing structures to improve existing functions of the proteins. For example, you can mutate 1 or 2 amino acids to improve protein stability. Fragment-based design use parts of existing proteins to combine them together in novel scaffolds and generate novel proteins. It can be used, for example, to create novel scaffolds, binding sites, and similar tasks. De novo design does not rely on existing proteins. So the general idea in de novo design is to create entirely new protein. It can be both sequence and structure, but the point is that you will not rely on existing natural information of these proteins. The complexity of the task grows from the rational design to de novo design, but de novo design has unlimited possibilities because you can create everything that you can. So after this brief overview of the physics-based and data-driven models, I just want to highlight that though currently many people focuses a lot on AI-based models, it does not mean that we should forget about physics-based models, because all of them have their pros and cons, and you just need to understand what are these pros and cons and then apply those models that work the best for you in specific tasks. For example, if you don't have many sequences or structures, or you don't, or you're not interested in a high throughput approach, you might still use physics-based models. But if you're interested in a high throughput approach or quicker predictions, then probably AI-based models are a better idea for you. Also, you need to understand the limitation of physics-based method and AI-based methods. One of the main limitations of the physics-based method is their dependence on template and scoring function. So if you have good template, existing molecule, and a good scoring function, probably you will get a good output. However, it will take a lot of time and computational resources. In contrast, AI models will depend heavily on the data quality and size. In other words, if your data that you're training your model on is biased or not accurate, you will have not accurate output. Another issue is that though the training of AI models also takes time, apart from it, another issue is their interpretability. For example, if you want to connect sequence to function, it's very hard to do with AI models because they provide you output but do not really explain how they get it. Now let's switch to more practical considerations regarding protein structure prediction and protein design. Let's start from the structure prediction. I separate the structure prediction tools based on what kind of systems they address. Here we can see monomer proteins, the protein that consists of one chain. We have multiple proteins that consist of several chains. We have non-protein system that consists, for example, of one RNA or one DNA molecule. We can have multimer complexes of both protein and non-proteins. For example, CRISPR-Cas that contains both RNA, DNA, and protein. And additionally, I want to mention special cases. In special cases, I put those systems which are a big issue for the currently available AI tools. For example, there are intrinsically disordered peptides and antibody-antigen complexes. Both of them are challenging systems, and currently available protein structure prediction models, no matter whether it's AI or not AI, struggle to accurately predict them, or at least require lots of time to simulate it accurately. The main issue with their complexity is their flexibility. As you can see, both intrinsically disordered peptides and antibody-antigen complexes, especially antibodies, has flexible loops or flexible regions. These regions are very hard to predict, and they require to take into account flexibility and dynamics that most of the available models do not have. But let's come back to more general models. I will talk more about AlphaFold3, Boltz 1, and Chai 1, because they cover the majority of the cases. So you don't really need to use many other models in everyday use. You can start from these three models to address both complex prediction, monomer prediction, RNA-DNA prediction, or other cases. Let's start from AlphaFold3. I will not talk about theory and some technical details because the next talk will be about AlphaFold, but I will focus more on the practical terms and the benchmarking. Here, you can see successful examples of AlphaFold3 predictions. Overall, I would say that those structures that have stable secondary and tertiary structure, and those biomolecules that relies heavily on evolutionary information, are very good systems for AlphaFold. AlphaFold can predict them very well, as you can see here. Such systems include, for example, globular proteins, soluble proteins, conserved proteins, or some conserved RNA, DNA protein complexes. However, when you don't have much evolutionary information, or when the systems are very flexible or disordered, AlphaFold3 cannot, or in most cases, cannot properly predict their structures. As I mentioned before, such systems include intrinsically disordered proteins or antibodies. So I would suggest you to be careful with predicting this kind of systems because the success rate is low and you need to be very careful with the output and check it twice. Overall, when we look at the statistical evaluation of AlphaFold3 predictions, you can see that it performs superior to the currently existing famous physics-based models or other AI models, and the overall quality is quite high and higher than the previous models. However, as I mentioned before, there is one case where the accuracy of AlphaFold3 is not high enough. This case is antibody-antigen complexes. To be more specific, based on the paper only 60% of antibody gene complexes were predicted accurately, meaning that even if you will try several rounds of AlphaFold3 predictions, 40% of your predictions will still fail or have not good enough accuracy. So please be very careful with systems with intrinsically disordered regions like antibody gene complexes. Another model called Boltz-1. Overall, AlphaFold3, Boltz-1, and Chai-1 are similar models for similar purposes. They differ a bit in the architecture and also in the licensing. So I will not focus on this detail, but just show that the accuracy of Boltz-1 is comparable to AlphaFold3 in different metrics. When we look at the Chai-1, we have similar evaluation. Chai-1 performs similar to AlphaFold3 in many cases. But one more important thing to notice is that Chai-1 might be better in antibody gene modeling than Boltz-1 and AlphaFold3. There is not a thorough evaluation of this statement in independent studies, but if you're dealing with antibody gene modeling, you might try first Chai-1 and especially Chai-1R. And if it will not work, try other models. Now let's move to the protein design. Here we will look into the protein design pipeline of a binder design. Usually we start from a target structure. This target structure can be a structure from protein data bank or a predicted structure. On this target, you will define hotspots. Hotspots is this place on the target towards which you want to design your binder. The next step will include generation of the general binder structure. You can use such tools as Diffusion. I think it's the most commonly used and widely used. So I will talk a bit more about Diffusion later. This step generates general backbone of the binder. And actually, you will generate thousands of them to make sure that a fraction of them will be successful. The next step includes sequence design. What does it mean? It means inverse folding that I mentioned before. Most commonly used tool for inverse folding is protein and PNN. So in this step, you try to fit different sequences or generate sequences that you can fit into the previously generated binder backbones. And the last step is in silico evaluation. Many people use AlphaFold filters like PLDT or IPTM or mole probability, which are the tools for structure check. But the important step that even after in silico evaluation, you will need experimental validation to really prove that your designs are accurate and functional. So prior to go to experimental validation, I would say to invest more into in silico evaluation, and you can be creative in this step by inventing more different types of approaches and scores, how to check if the design structures with sequences make sense for your task or not. And this design pipeline is Bindcraft, a widely used method to combine all the steps of the typical protein design pipeline. It's open source and you can find it on GitHub on this link. I'll share the slides later. And as I mentioned, I will focus a bit more on Diffusion. Diffusion is a generative model that can generate different types of protein designs, not only binder designs, but different applications in general. It is based on RosettaFold, and RosettaFold is a structure prediction model similar to AlphaFold. So you can also try to use it for structure prediction, but based on my experience, it's slower than AlphaFold and other models, so maybe it's not the best idea right now. So Diffusion can generate symmetric oligomers, for example, or it can generate binders, as I mentioned before, when you provide the target with hotspots. Another important thing is motif scaffolding. When you, for example, have a part of the structure that you want to keep, and you want to generate the remaining structure around this. It can be especially important for enzyme design, where you have a catalytic site which you want to preserve, but you want to redesign the surrounding of this catalytic site. Then you can use Diffusion with motif scaffolding and symmetric scaffolding of general structures. As a conclusion, I would say, out of all available AI models, and there are thousands of them right now, if you are interested in structure prediction, look first into AlphaFold3, Boltz 1, and Chai. Try them, and in most cases, they will be enough for you, except when you're dealing with special cases like intrinsically disordered proteins and antibodies or antibody-gene complexes. For protein design, the most used tools are RF diffusion for protein generation, Bancraft of the whole pipeline of protein design, protein binder design, and protein NPNN for sequence redesign and inverse folding. As I mentioned before, challenging systems include those systems that involve disordered and flexible regions within. Also, another types of challenging systems are those that are currently underrepresented in protein data bank. As I mentioned before, AI models and data-driven models in general heavily rely on the training data set. So if there are some types of systems, for example, lipids that are underrepresented in the training dataset, then the model will not be able to predict them accurately. And to understand what actually works well and what not, you can just look into the distributions of different types of biomolecules in PDB, because most of the structure prediction tools were trained on PDB, and see which classes are underrepresented, and then most likely they will have less accuracy in the output. Though there are exceptions when the model is able to generalize over different classes of biomolecules, but you can still use the training data as guidance for with what model can deal with better. This is all from my side. I think I am a bit quicker than my time, but maybe we'll have a bit more time for the question sessions. And I will share my slides in the Slack after the talk. Thank you very much for your listening.
Super. Thank you very much for that lovely talk. Do you have any questions? Okay, I could pose one to get us going. You talked about Chi-1 and how it was slightly better for modeling antibodies. I was wondering if you could maybe tell us about the scale you need to model on to sort of get it to make antibodies accurately. I think I recall AlphaFold3, you needed to make lots of models, so I was wondering if it was the same for Chi.
Yes, it's a general trend that you need to do lots of models to be able to get an accurate prediction. And by lots of models, I mean thousands. But to be honest, I feel like even though Chi-1 claims that they outperform Boltz and AlphaFold3 and antibody-gene modeling, the results on the practical testing actually quite similar. So I would say the best idea to approach antibody-gene modeling is still use physics-based methods that involves molecular dynamics that are able to properly and accurately model the fluctuations of the binding loops of antibodies. And this will give you the most accurate results.
Thank you.
We have a few questions on Slack, so I can ask the first one. So most of these AI structure prediction tools failed for larger proteins, meaning more than 150 kilodalton, or input sequence of of 1,000 amino acids, or more than 1,000 amino acids. Which model is best to predict structures for those big proteins?
Well, there's nothing better for multimeric and big complexes than Chi-1, Boltz 1, and AlphaFold3. So if these three models will not be able to process biostructures above 1,000 amino acids, probably all others will fail. And the issue here is not exactly in the models, is computational resources needed to make the proper prediction, proper training for this kind of molecules. Plus, there are in general less of them in the training data set, so it's harder for them to predict. One suggestion that I may give you is to try to simplify the system first. For example, many of these complexes might be oligomers or homoligomers, which is even better, that consist of similar subunits. And then you can first predict the structure of one subunit and then model them together. It can reduce computational costs and increase accuracy because in general, both physics-based models, physics-based tools and AI models are able to predict more accurately smaller proteins and complexes. So I would start from getting a subunit and then try to predict it and then rearrange them together as a complex, if you know how in general they look like.
Thank you. I can add a bit on AlphaFold part. I am Oleg Kovalevsky, Google DeepMind. So it mostly depends on amount of RAM on your GPU, really. With AlphaFold3 on 80 gigabytes 800, you can model up to 7,000 tokens, which is about 7,000 amino acids for protein-only structure. For some large targets, we saw a little bit of degradation of quality of prediction after about 2,000 tokens. At the same time, we had very successful examples of modeling like 6,000 tokens. So it really depends on case-to-case basis. And please pay attention to the confidence scores. They will clearly tell you whether modeling is running well or AlphaFold is not doing a good job. On AlphaFold server, you can model up to 5,000 tokens.
Okay, do we have any more questions? Oh, one more.
Hi, Jos Kams, ESRF. Do you know if there's anyone looking into or any tools that you could use to look at interfaces between homodimers, and whether anyone is looking into how you can either increase or decrease the stability of the homodimer, for instance?
Yes, there are many models for protein-protein interactions. I think they're more general, like for any kinds of protein-protein interactions, but probably stable protein-protein interactions. If you tag me on Slack, I can send you the links to the papers and tools.
Okay, thank you.
Just out
Okay,
of my
I
mind,
think-
there is one general tool, it's called Massive, that I think is quite famous for protein-protein interaction. And you can start from this one, but it's quite old already. There are much more tools right now for these specific purposes.
Okay, I think we can move on now, but I'd like to thank Eva again for that talk.